let me remind you of the few events that are left. So we have 4 meetings on Tuesday. Bewilderingly will be giving a talk on where the philosophy of cognitive science went wrong, so the philosophy of Coxside. Brice will be arriving, I think, on Monday. And so if you want to talk to him on Monday or Tuesday, please let us know. On Friday, Laura Gradowski, who is a postdoc with us, giving a lecture on expectation and exception, expertise as an epistemic constraint. We are looking forward to that talk. And after that, the following week, we'll have one event left, where Dan, who is here, will be battling with Wen Wu from the CMU on the topic of intentions, if I remember correctly. Is that right? So we'll have a debate and an exchange. Each will have 20 minutes, followed by a 10 minutes response. No blood, no weapon.
Umbrellas.
Maybe umbrellas.
You haven't made any agreements about that.
Oh no, we don't make, but I'm laying the rules.
Oh,
So it's at the semester we'll be done.
You're from the south.
All right, and today it's my great pleasure to introduce Devin Gouvêa, who many of you know because they just moved us for the whole year. She was a visiting scholar last semester and a visiting scholar this semester. Devin is an assistant professor at the College of the Holy Cross. She got her PhD in Chicago. She works in the philosophy of biology with an interest in homology, but also she also works in the general philosophy of science, mostly with a focus on scientific concepts and how they change or how they fail to change and why, and the rationality of conceptual change. It's been really a pleasure to have Devin with us this year. And I'm not exactly sure whether the talk would be on the same topic or a different topic. But we'll find. Okay, so another area of research, as the topic of the talk is crafting scientific narrative. Floor is yours.
Thank you. Time here. Great, everyone. Thank you for coming. And I particularly enjoyed preparing this talk because, as Edward anticipated, looking at my title and comparing it to what I've been talking about before, this is something different. It's a project I actually set aside about 5 years ago when it was time to really focus on my dissertation and actually finish that PhD, but it was a lot of fun and it's been fun to pick it up again. So I'm really curious to hear your feedback, what works, what you like, what you don't, so I can kind of decide what to do with it. So I'm going to address this topic of how scientists craft narratives, hence the title, mostly by giving you a narrative of how this unfolded in a particular lab in which I was situated. So there's a lot of like meta layers of stuff going on here. Some motivating questions that are behind this project. Kind of the most conventional established philosophical question is this one that often goes under the heading of data interpretation. So how did data, observations, measurements, readings, whatever, become evidence for some particular scientific claim? And I think the earlier literature on this topic focused a lot on how, like, sort of what is the justification of the inference from data to evidence to claim. Now there's been more interesting look at sort of how does this particular data match up with this particular claim? Is that flexible? How does it work? There's sort of a reflexive motivation here. Like, why is science so hard? And in particular, like, why was it so much harder than I personally expected to produce the scientific paper that's filling with mine here? So this hat is right. So I wrote the scientific paper as part of the requirements for a master's degree in evolutionary biology, which I was doing alongside my PhD at UChicago. And it took longer in the lab than I expected, but the really striking thing was how much work went into crafting this narrative, sort of starting with all these experiments that I did and producing this story that could be published. And it was a very iterative, kind of time extended process. So that's underlying there. And then this kind of blue sky question, which is just like, can we as philosophers of science talk about creativity? Can we talk about creativity more? Is that like in the context of discovery and it's only for the psychologist to consider? So this is something I think we should be able to philosophically engage with, but I don't really know how to do it. So I'm just kind of throwing that out there and I'd love to hear your thoughts. Okay, so here's the table of contents for my narrative. As you see, there's kind of going to be 3 stages of the main narrative of the science. We're 2, 4, and 6 here. I'll introduce the dramatis personae, the characters, both human and non-human, sort of the plots of the research, and then kind of look at narrative formation on 2 scales. So like this big research trajectory. The benefit of having 5 years since I last worked on this project, is the lab has done 5 years of work. So I can now see this like nice decade-long change in all the stuff that they've been doing. And then the smaller scale of my individual publication. Kind of interspersed with those stories, I'll have these little philosophical interludes on topics of narrative, phenomena, and data. So things that I just think are helpful resources that I'll kind of refer to throughout the talk. All right. So I'll start with the first interlude on narrative. The most important source of work on this, I think, has been this narrative science project spearheaded by Mary Morgan, who kind of did some of this work individually, expanded out. There's a recent book here with a bunch of great case studies on narrative science, special issue back in 2017 in studies. So lots to explore there. And I think one of the things I like about this project is they've taken a very sort of open conception of what narrative is and explored kind of all the different ways that it can show up in science. And it's really quite amazing, all the different kinds of cases here. I'm just going to focus on one contribution to that volume that I just showed on the previous slide by Robert Meunier, who's been with this project for a while. And he introduces actually drawing on this kind of work in theoretical narratology, which is apparently like a field. He introduces a number of really helpful concepts and kind of that I think are useful for charting the way through my project. So the first is a distinction between narratives of nature and research narratives. So narratives of nature are kind of the things you find in textbooks or nature documentaries, like, you know, this enzyme does this thing in this place, or this animal mates in this season or whatever, right? These kind of general things that happen even when the scientists aren't watching. Research narratives are the things that we publish and produce in our paper, but there's an important connection between these. So Robert's going to argue that sort of by bringing someone through a research narrative, the scientist makes it possible for people to see how those narratives can contribute to the narratives of nature, how those two things inform each other. There's also sort of a transformation in this narrative generating process from the authors, who are the actual scientists, to the sort of narrator as character. So in our paper, there was kind of this sort of we narrator character, who is also a character in the story, who sort of did certain kinds of things. There's also a distinction and a transformation between this practice world, where all the experiments got done, and then the story world, what we tell in the paper, which there's a relationship between them, right? The practice world very much constrains the story world, but the order is not the same, the emphasis is not the same, the details are not the same. So this is a point that's been made before this narrative work, that there's a mismatch between what's in the scientific paper and what actually happens. I think thinking about it in terms of narratives gives us a sense, a way to start to try and understand why that happens and how it can be useful. So the purpose of the research narrative, Robert says, is to represent the practice world events as an approach to a given problem. So instead of just giving all of the nitty-gritty details of what we did, to sort of recognize we've got this general approach, it allows us to understand these things and not those things. And then also to create a logic of how the different practice world events hang together, how they may be causally connected, how they illuminate each other. So the mismatch can actually be kind of virtuous. There'll be lots of evidence of mismatch in this. Okay, so let's talk about the science a bit, the key characters. The main sort of star of the show here is the worm, Caenorhabditis elegans. So one of the articles from which I got this top image, and I should say just inside this top image is the connectome of the worm. It's sort of how all the different neurons are connected, and it comes from a computer model that is called Open Worm. That shows all of these different things. So you've got this computational model of worm biology. And an article that I read about this called C. elegans, the world's most understood multicellular organism. And I think this is true. It's pretty simple. It's got less than 1,000 cells, about 300 neurons. But we know how each of those cells can trace back to the single-celled embryo. We know how all the neurons are connected. We've got the genome. There's a lot of what we know about this organism, and yet a lot that we're still trying to understand. So at the opposite end of the spectrum from this computational model is sort of what it actually looks like where the worms live in the wild. So they live in kind of anywhere that rotting fruit can be found. So figures E, F, and G there are sort of like the micro scale of where you might find these worms. They're about 1 millimeter long, the adults, and they share their rotting fruit environment with lots of lovely critters, which you can see in panel H there. Those are spring-tailed, those little white blobs, I guess. Then in I, that's kind of what you see under a microscope when you work in a C. elegans lab. And here in particular, these are worms crawling out of fruit. So there's been a lot of work in recent years to actually cultivate natural strains as opposed to the laboratory strain, which has been propagated for decades and is different from the wild strains and various. And then also these poor worms in the wild are sort of infected with all kinds of parasites. So this is like a worm snout with a bunch of fungi growing out of it. So life in the wild is much tougher than life in the lab, basically, is what you should take away from it. Okay, so worms are important. Oh, right. More about worms. So they have two sexes. There's a hermaphrodite that first produces a bunch of sperm, then produces oocytes, and then a male that just produces sperm. In the lab, C. elegans males are really rare unless you stress the population a lot. So one of the interesting questions is like, why do they stick around? They're not necessary for reproduction. And some of the work that we'll look at later, we'll talk about that. Here's a closer look at this, just the reproduction by self-fertilization, which is kind of the standard mode for laboratory populations. Super useful for laboratory reasons because you can get these like genetically identical worms, right, over time. They're just fertilizing themselves. It's great for lots of reasons. So kind of zooming in on the middle of the worm, there are these gonad arms. The cells start getting formed here, and then they kind of progress around here, eventually get cellularized. And early in the worm's development, a bunch of those cells coming around the bend turn into sperm. Later in the worm's development, kind of until the end of its life, they turn into oocytes. And once the sperm are exhausted, the worm can't produce any more embryos. It just sort of pops out. All right. Oh, and I should say, this is a good point to say, if I say anything confusing about worm biology, please feel free to interrupt, because there's a lot of science in the topic. I want you to be able to follow it. Yeah. Yes. What about it? Where is it? Did you say?
No, but there's a.
Oh, there's an embryo in here. So.
Worms have uterus.
They do. They do have uteri. Yeah, small. But there. Yeah, so the spermatheca is where the sperm hang out. And then like if you were looking at a more fully developed worm, you'd see like maybe 5 or 6 embryos stacked up, and then they kind of lay them in little bursts and then keep going. Other questions? All right. Okay, now we're getting to the people, right? So the Ruvinsky Lab, this is the lab that I worked in. It's at Northwestern now, it used to be at U Chicago. Ilya Ruvinsky is the PI. He was my advisor for my master's degree. And then Aaron Aberson has been a staff scientist since before I arrived in the lab. So maybe 12 to 15 years now. So these two are kind of like the mainstays of the lab. And other sort of students and postdocs that can come in and out. So note the tagline here. I don't know if you can read it, but under the lab title, it says discovering pathways of social signaling. Okay, that's kind of like the way the lab describes itself right now. What does that mean? You can get a sense by looking a little more closely at sort of some recent publications. Right, so what social signaling that does what? That regulates development and aging, and then the neuronal circuits that are involved in that. And if you look at the paper titles here, you can start to get a sense that the social signals here are really social signals in the context of reproduction. Okay, so we have a male pheromone that improves the quality of the oogenic germline, right? So this is pheromones released by those males that aren't super common that act on the oogenic germline of the hermaphrodites and affect the quality of the eggs. Serotonin coordinates this response. So in some recent research, it turns out if you feed worms Prozac, it affects their germline positively. It's definitely reproductive lifestyle because these neuronal circuits are controlled. Metabolome, this is the collection of all of the small molecules that the worm produces, both inside and outside. So the pheromones are part of the metabolome. These are different between the two sexes. So this line of research here, these are just the most recent papers. It's been really fruitful right now. It's producing a few papers a year in a pretty small lab. So that's great. And they're really kind of getting down to a lot of the mechanistic details of what's going on in these effects. But the lab hasn't always studied these particular things. So this is kind of another plot line, a couple of other plot lines that were in lab when I joined it. So that paper highlighted up there at the top is my paper. And this is sort of a shorter plot line focused on, again, the reproductive system, but particularly on stress and heat stress. So different from the social signaling kind of thing, right? Looking at the same bits of the worm, but looking at it from a very different perspective. And then before that, the lab had been focused on a lot of stuff involving genes, genomes, gene regulation. So this stress, I arrived at an interesting time, basically. The lab was kind of, genetic work was winding down. They were looking for some new stuff. And there were these interesting papers on stress in the reproductive system, which as I was leaving, shifted direction in an interesting way. So how do we get from one plot line to another. How do we get from this first paper in the reproductive stress response category to one of these most recent papers in the social signaling category? So that's what I will focus on in the next part of the talk. But first, I want to do another philosophical interlude to talk about phenomena, because I think a lot of this story is helpfully told by tracking the different phenomena that the lab is focusing on at different points in time. What are so probably the most influential line of work on this is by Jim Bogen and Jim Woodward in their pre-Pittsburgh days. And this work was focused on sort of challenging an older view of confirmation or justification in which data sort of just directly informed theories or theories predicted data. Instead, they argued that there's this kind of intermediate epistemic stage of identifying phenomena, which are maybe not directly observable, but kind of transcend labs, these sort of interesting stable regularities. And so those are the things that theories predict and explain facts about. And so then a lot of the interesting questions about justification and confirmation and avoiding errors and artifacts and stuff like that happen in this data phenomena relationship. So that's a really important development, kind of a little bit earlier, but for different reasons. Ian Hacking is also thinking about phenomena, but focusing on a different aspect of it. He's focusing on how hard it is to get phenomena in the first place. And I love this passage. If phenomena were plentiful in nature, summer blackberries just there for the picking, it would be remarkable if experiments didn't work. But in fact, it's hard to get phenomena. It's hard to stabilize them. It's hard to make your equipment work. It's hard to decide what equipment you should use in particular situations, depending on what you want to know. And so phenomena involve a lot of effort, a lot of skill, sort of creative work. And that is a hard task. And in fact, he says there's multiple tasks. You have to sort of figure out when, like I said earlier, when are the experiments working? How do you know? So on and so forth. So one more development in this phenomena story. Uljana Feest in 2011 tries to sort of bring Bogen and Woodward and Hacking's idea of phenomena together. So she's trying to bridge sort of validation, making sure you're not making an error kind of aspect of phenomena data relationships, and this skill aspect, both physical and conceptual skill of figuring out what to study and how. And so for her, the surface phenomena, she argues that there's not just one thing that is a phenomenon, basically. There's sort of this interesting iterative relationship between the surface phenomena, those are instantiated by the data, and they, for her, are these empirical data patterns that you either find in the world or you create in the lab. So they're not just individual data points. There's a certain level of regularity, of abstraction, maybe different labs can create them. So she argues that they have some of the characteristics that Bogen and Woodward think phenomena have. But at the same time, what the researchers are really interested in are these more hidden phenomena. She doesn't necessarily want to say that they're unobservable, but they're at least harder to access. So things like, the responsiveness of this part of the worm to this signal from males or something like that. That would be a hidden phenomenon that we can't directly observe. We have to get at through these surface phenomena of patterns in our data. So she argues that when you're trying to stabilize phenomena, you're trying to kind of say, this is a thing that we've got that we can use to explain whatever it is that we want to explain or predict things. What we're really doing is kind of adjusting the fit between these surface regularities that we can see in our data and these sort of underlying hidden ones that we see otherwise. So we might, go back and forth thinking that we've got some underlying process, realizing that we're wrong because our data says this, getting more data, and eventually stabilizing. Okay. So I'll be drawing on some of these ideas of phenomena in what comes next. Now I'm going to return to this question of how do you get from talking about reproductive stress response to talking about male signals over a period of 10 years. So I'm just going to call this scale A because there's probably a lot of different scales at which you can analyze this process, but this is going to be an extended research trajectory. All right, so again, how do we get from this first paper where you're modeling stress response? I'll say a little bit more about the sense in which they're modeling stress response in a minute, to this communication between the male pheromone and the oogenic germline. Okay. So diving in just a little bit into this 2012 paper, which was kind of just being finished up when I first got in the lab and started figuring out how to touch worms without killing. Total novice, right? Needing to learn lots of those skills that Hacking is talking about. So this paper came out of a collaboration between 3 labs who all worked with C. elegans for different reasons. And we're all interested in kind of a key question in systems biology as they understood it at the time, which is how do you get from tons of complicated molecular interactions that you're trying to study, sometimes with huge amounts of data, to these sort of basic organism level behaviors. They were really interested in whether we could model something, some important behavior that we maybe knew a lot about on the molecular or the genetic level in a very simple way. So could we find a behavior that we could model as just the interaction of a few key components? And the hope was that finding a successful model like that would help us figure out kind of what to focus on among all of the molecular mechanistic data that we have. So they tried a few different behaviors, and then they focused on egg laying at different temperatures. So the basic paradigm here was take a bunch of worms, synchronize populations, put them on separate individual plates so you can keep track of their output. And then when they're just about to start laying eggs, so like they look like that schematic worm that I showed earlier with right before that first embryo shows up in the uterus, you shift them into different temperatures, and then you just let them keep laying eggs and count, and they'll lay about 300 at sort of normal laboratory temperatures. So they trained this model at a couple of different temperatures and found that they were able to predict. Okay, great. This was exciting for their own modeling purposes, but it also kind of sparked future interest. So you'll notice that that blue line there, 23 degrees, right? Pretty steady accumulation of eggs, right? Once you get up to 28 or 30, really not much egg laying happening. Okay, so the temperature, the reproductive system is really, really sensitive to temperature. So I mentioned this paper in that level of detail because it seeds that interest, but also because there's some features here that become really important in where the lab goes next. So they focused on something that's ecologically relevant as a phenotype. So egg laying, super important in like the worm's general function. And I say ecologically because temperature is a thing that varies a lot where worms live, not in the lab where they just hang out in the incubator, but where they live, it varies a lot. I'll come back to that in a second. So here's, for instance, like natural temperature variations in the summer where we know that C. elegans has been recovered from Lisbon, right? The temperature's all over the place, and it's in that range in the black box where we know that egg laying kind of tanks, right? So worms are clearly experiencing temperatures where their reproductive systems aren't happy. How does that work? Okay. And the other thing the lab is doing is focusing on individual worms. I mentioned that they're tracking each one, how many eggs does each one produce. And that's going to be really important for kind of all of our future experiments, that you can catch different kinds of phenotypes when you are just looking at individuals. Okay, the last thing I want to say about this ecologically relevant thing is that before the work of our lab, kind of starting with this paper, there was really sort of a gap in temperature studies between kind of the fertile range where you can just keep worms their whole life and you can maintain populations, and then heat shock where you would just put them at almost lethal temperatures for a few hours. So we know a lot about what happens here, and we know about heat shock. There's a lot of cool machinery that the worm has, but not really much was known about sort of like 12 to 24-hour exposures in this like uncomfortable range. So that was what we ended up focusing. All right. So that's the starting point. Here, zooming out quite a bit, is sort of stages in the evolution of the different research questions. So I talked a lot just now about how we can model macro level physiology as the interaction of some few components. That's what we did. And then having been sort of sensitized to the effects of temperature on the reproductive system, the lab shifted into questions about how heat stress affects reproduction and why, like in the distinctive ways that it does affect reproduction, how might those have evolved? Which then, the second green question here is the one that I ended up focusing on. How does experience affect stress response? So particularly the experience of prior stress, right? So if you've already experienced stress at some point in your life, does that affect your ability? Or your parents have experienced stress, this is what's going to happen in the wild. So we got interested in that. Then through mechanisms we'll talk about later, they sort of jumped to this question of why C. elegans still has males. What are males doing for the species as a whole? And then eventually these more mechanistic questions of how male signaling affects hermaphrodite physiology. Okay. So, right. So this is a, gives you some sense of how it changed, but it's still kind of opaque. So as I was trying to kind of get a better handle on how this transformation of questions happen, I found that it was really illuminating to look at the interplay between phenomena, right? I talked before about how figuring out what the phenomena are is not a straightforward process, and it involves a lot of iterating between different things. But here I'm talking about a further level of iterativity between the phenomena and the procedures that we use to study them. It turns out that those questions, which might look a little bit random, ordered in that way, start to make more sense if you see how we started studying this phenomena and we used this procedure, and then that led us to this other phenomenon, which led us to this other procedure, and so on. So making that slightly more concrete, I already talked about this macro level behavior, this general interest in that phenomenon of, you know, what does the worm do at organism scales, which led the lab to do this counting of egg laying over long periods of time, which led to an interest in the reproductive stress response. So not just laying eggs, but like particularly, can you produce baby worms that can then keep the population going, right? So in future studies, mine and others will talk about, we then started looking at live offspring. So after you're at stress for a period of time, then you go back down to a happy temperature. Do you produce new worms? Does that happen? That, through an interesting serendipitous development, led to a focus on sexual signaling, which then leads to a very different central measurement of counting these things called germline precursor cells. Doesn't matter that much about what they are, other than that they affect like how many reproductive cells you have and how long those last during your lifetime. So if you're looking at the effects of these chemicals on the reproductive system, that's this great, like really precise, reproducible thing. You can just throw a bunch of different treatments at the worm and see how the gene cells respond. Okay, so this transition here with the red arrow from focusing on live offspring with an interest in temperature stress to talking about sexual signaling is one that is especially significant. So I'm going to talk about that now. It's kind of a turning point in the research. And then I'll get into my little contribution to the reproductive stress response. Okay, so because this is an important experiment, I'm going to go into the details just a little more. Kind of already hinted at this. But now we get to look at some petri dish diagrams. So L1 is basically just the first stage of development after worms hatch, and there's sort of standard procedures in the field for getting a population that's in developmental arrest. It doesn't have any food, so they're just kind of hanging out. But they're all at the same stage, right? So if you put them on food, they start growing and you get the synchronized population of young adults. So that's all standard. That's great. What was less standard was this idea of singling all of the worms and then paying attention to what they're doing. So you sit there under a microscope with this little platinum pick. This is what I had to learn how to do, my first skill, and then separately kind of move them to new plates without killing the worms or digging holes in the auger. Then we'd stick them in some sort of uncomfortable temperature for 24 hours, take them out. Okay, so now we still have these individual plates. We count eggs. This is kind of a holdover from what the lab had done before. We were particularly interested at this point in like, can you regenerate the population after heat stress? But we kept counting eggs, and it ended up being useful for learning other things. So who, you know, basically how many eggs have they laid during stress? And then you wait a few days and see if they're live offspring on the plate. Okay, so this procedure here that I just unpacked, I did spend a lot of time working with, other people spent a lot of time working with. One thing we found was that at sort of 29 degrees, so that's about 86 Fahrenheit, 29 degrees, not super, super hot. Reproduction, recovery of live offspring is really low. So this first bar here is kind of from a previous paper. Only about 20% of all of the worms that go through this 24 hours heat stress are going to come out on the other side and be able to produce some sort of live offspring. So that was something we looked at in other work. But it turns out that if you stick a male on the plate with the hermaphrodite during the heat stress, they suddenly, a much larger fraction of them are able to recover, like 3 times as much. And then it turns out you don't even really need the male. You just need whatever scent the male leaves behind while it's crawling around on the plate, right? So you could let these males just kind of condition the plate for a while, take them off, stress the hermaphrodites, and they're still going to recover almost as well. Hermaphrodite scent really doesn't make that much of a difference. These other mutants here, they're important because they affect these pathways for producing the pheromones, right? So depending, the details don't matter, but kind of if you manipulate the pheromone pathway in various ways to change what pheromones the males produce, then. And then again, it turns out you don't even need the male scent. You can just put like a special combination of pheromone chemicals on the plates and it smells like a male, and then the germline responds in all of these really interesting ways. Okay. So this whole paper, this was a 2015 paper, so it was kind of happening as I was finishing up my much less exciting, I will admit, paper. But it just kind of came out of a sort of spontaneous situation in which my coworker, Erin, who I mentioned before, was doing a bunch of experiments, not with males, just with hermaphrodites, doing that same stress procedure I talked about, looking at wild isolate strains. So not the laboratory strain that everybody uses, but strains that had been more recently pulled from Hawaii or Lisbon or whatever, and seeing how they recovered. And she was really bored. And so she just put some males on the plate one day. And lo and behold, they recover much better. And they were sort of interested in males. Like people would always ask, and the lab would give talks like, what about the males? Why are the males there? And we don't really know. So she did this. And here we go. So I guess one other observation that's important to mention, some of those wild isolate strains produce more males, right? So that gave us another clue that maybe males were in fact doing something interesting. So from here, the lab just kind of has run with this, focused on this, decoded it mechanistically, all kinds of really interesting. And so now we get papers, for instance, so this orange here, ASCR number 10, ascaricide number 10, that's kind of like one of the key pheromones that's involved in this process, and we're breaking down when that pheromone affects the worms. Now we can see, we don't even need to look at something as complicated as recovery after stress. We can just see how many of these precursor cells there are. And so here, these are all like young worms, basically, like before they would start reproducing. They're not that sensitive. Over here, you can see there's big differences between the collections of gray dots and the collections of orange dots. So if you have this pheromone around, the worm produces a whole lot more of these cells that are going to produce future. And there's all kinds of interesting implications for trade-offs between investment in your germline and investment in your soma or the rest of the body, right? So it seems like basically at a big high-level evolutionary perspective, what's going on is the males are sort of adjusting the way in which the hermaphrodites contribute their resources to their germline as opposed to their soma. So this has, some potentially harmful effects for the hermaphrodites in the short term. There's some lifespan costs and things like that. It is, however, very helpful to have outcrossing when the population is under stress. It's helpful to have more reproduction with worms of different sexes rather than just self-fertilization. So there's trade-offs on all kinds.
Devin?
Yeah.
If there's a male in the dish with a hermaphrodite, is there fertilization from the male in addition to the hermaphrodite? So you just said there was outcrossing, so it's
Right.
reproduction.
Yeah, so in the wild there would be, but in these experiments, that's not what's going on, right? We're making sure that they're not, we're just looking at the way the males affect the future self-fertilization of the hermaphrodites. So yeah, obviously that having fresh sperm is helpful, we know, for the worms, but we're interested in, like, even without mating, right? Like, what do the males contribute that's helpful, or how do they affect? Does that clarify?
Oh, yeah.
Okay, cool. Other questions? All right. Okay, so that's our big story. When we sort of look at this kind of really interesting interplay between data, phenomena, research questions, procedures, we see that the research can go in really unpredictable direction. The last thing I'll say before I move on to my story via the data is that it also made sense for the lab once they realized this connection to pursue the social signaling. The phenotypes were just easier to manage. The effects were bigger. There were more resources for breaking it down mechanistically. So the lab couldn't have said, oh, let's go study social signaling now, until they had this sort of spontaneous result. But once it appeared, it made sense to follow it. So there's some creativity here, but also a kind of like sort of strategic calculation about the best way to go. All right, last philosophical interlude, because I'm going to be talking about on the smaller scale, the data in my project. Here I'm going to be just saying, drawing on key things of Sabina Leonelli's work. Lots of cool stuff in recent years. So she has this relational view of data, which I think is really helpful for understanding this case and other ones, where she wants to argue that data are not sort of just intrinsically representative of certain things out there in the world. The way in which they can represent or document different things depends on the situated ways in which data are handled. So basically, like, where they're being used, alongside what other data, by whom, with which tools available, right? And so if we want to understand how data represent, how we interpret data, we need to look at their situation. She also has this concept more recently of data journeys and data as lineages. So there's kind of like two metaphors in here, both of which I think are helpful and probably will be developed more in the future. So the idea of data as lineage is just that they're things that transform over time. They have a history. They start out perhaps meaning one thing, end up meaning something else, kind of maybe having new measurements added, change happening. So she said these are objects that need to be transformed. So that's the lineage part, in order to travel and be reused for new goals. So the travel here is traveling in space and time, maybe from different perspectives. But most of the work, this is another case study book, like the scientific narrative book. And most of the cases are things where data is traveling from one lab to another, or from one research context to another, like across large distances, maybe through computational databases, and often, but not always in these cases, in the form of big data. And so I'm taking this idea a little bit out of context and arguing, or suggesting, not ready to argue much yet, but that even on really small scales, data travels. Maybe it takes a walk around the block, if we want to say, kind of within a lab. It's, these are not like big journeys where you're packing your suitcase and you're going to France, right? It's just like, okay, the data meant something when we produced it, and then maybe it meant something else later on. So that's what I'm going to be talking about next in this bit of the thing. So now we're talking about narrative formation on a small scale, single published article. So like I said, this is a reflexive case study. I'm in part trying to solve my own problems here, about why this paper was so hard to produce. And again, just to remember, the key experiments I did was this procedure I mentioned before, where we take synchronized populations, let them grow to young adults. Then these are all hermaphrodites here. I wasn't working with males at all. Then we stick them in heat stress and see who recovers baby worms after 5 days. Okay, so what I want to point out here is the way in which stable data patterns, stable regularities, these surface phenomena in the Uljana Feest sense don't really mean very much on their own, right? Okay, so I showed this in a different context before, but we know that if you raise worms at 20 degrees, shift them to 29 degrees for 24 hours, and then shift them back down, about 20% of them will recover by offspring. So here we have like 6 trials. Like this is a pretty stable regularity. Other labs could reproduce it. So it's not just, one data point. It's something like a small phenomenon, but it's not very interesting on its own. Like who cares that 20% of worms raised to 20 degrees and stressed to 29 degrees for 24 hours? Who cares, right? It only becomes interesting when you put it alongside other data, other surface patterns. So now we have a slightly more interesting data pattern. So this is, and it's not my paper, but this is the paper that just preceded my paper. So my coworker, Aaron and Ilya basically wrote this paper. They were working on it, but they had found this big result when I arrived at the lab. So I want to focus on this red curve here, this recovery of fecundity, which is the procedure I was just showing you, where you see who makes babies. You would think from sort of just intuition that as it gets hotter and hotter, that fraction should go down over time. But in fact, you get this really weird spike at 31 degrees. And every time I presented this for a while, people were like, well, did you do something wrong? Like what happened? No, like this is a very, very stable regularity in the data. Okay, so it turns out basically that what happens is that at 29 degrees, basically all the way up to here, the worms keep laying eggs during stress. And this is really bad for them. Kind of, they waste sperm, they sort of gum up their gonads. And that's part of why when they come back down to a normal temperature, they don't reproduce very well. At 31 degrees, all of a sudden, the animal is like, no, we're not going to do this. They just don't ever lay eggs. They don't ovulate at stress. They just sort of pause until they get out. And that turns out to be really productive, right? Like when the stress is over, if you haven't used up any of your sperm, you haven't ovulated, great, you can go ahead and produce offspring. So then you ask, like, well, why don't they just stop sooner? I don't have the figure for this earlier, but there's a trade-off between if you totally stop your reproduction, it takes some time to start it again, right? And you don't know how long the stress is going to be. Like we know the stress is going to be 24 hours in this experiment, but worms don't know that. So it turns out at shorter times, like 12 hours, even though ovulating at 29 degrees is bad for your reproductive system, it's a better strategy because the stress is over. You don't have the pause period of like waiting to catch up, right? So there's sort of like a bed hedging thing that's happening here. And it's really interesting, kind of, you thought, maybe we can learn something about how this evolved. So that's kind of where we were when I showed up. So all of a sudden, this boring regularity, which is right here, has become a lot more interesting. We can construct a story around it. It's great. Then I started trying to build more things around that regularity. So we decided to look at the effects of growth temperature. 20 degrees is just this arbitrary temperature at which we keep all of our incubators. The populations grow well, but it's 68 degrees. Worms live in lots of places that's not 68 degrees Fahrenheit. So we looked at rearing worms at both colder and warmer temperatures, and then doing the same procedure. That's kind of was the heart of my project. Eventually, when we started, we thought, oh, maybe there'll be this like beautiful, nice shift of the curve, and we can somehow understand something mechanistic about what's going on. No, unfortunately, it was just kind of messy and confusing. So I spent a lot of time juxtaposing these curves, making other curves, trying to make sense of them, trying to figure them out. This was a lab meeting presentation around 2014. And it only became something that we could kind of do something useful in building a narrative with when somebody in the lab meeting suggested, like, why don't you stop making curves and maybe just pick some of the most interesting things to focus on? And this was a shift. It seems obvious in retrospect, like there's too much going on here. It's not useful. But thinking in terms of curves is what made the last paper work so well, right? So we had to change how we were narrating. So kind of a creative movement that happened. And so now we get this thing that became kind of the focal phenomenon of my paper, which this is the same data, just a subset of the data that was in this previous thing. So it's like this little line of dots here. But immediately you can tell once we switch to bar graph, it's a lot easier to see like, here's another interesting pattern, right? Like being grown at 20 degrees for this particular stress paradigm is worse than being grown either at colder temperatures or at warmer temperatures. Then that was something that we could then try and explain. And so basically my paper and my work ended up being trying to explain what was going on here. I won't tell you the whole story, but basically there's some effects that happen at 15 degrees that are helpful, and there's different effects that happen at 25 degrees that are helpful. 15-degree ones involve like the germline itself and the physiology. The 25-degree ones seem to involve the actual embryos, like the mothers are sort of aware in some way, chemically aware that it's hot, and they do something to the embryos that we don't know what it is. So these two totally different effects. Okay, so again, not going to go through all of this, but this panel right here is that phenomenon. So we were now able to juxtapose it with a bunch of other things, laying behavior, who hatches, who doesn't, and start to construct a narrative around what was going on here and get some explanations, not as definitive as we'd hoped, but some explanations of this phenomenon. So here I think we're seeing like these data are traveling around our lab and the phenomena are being adjusted in our lab in interesting ways to make this narrative. Okay, then that one figure is now in the context of a whole lot of other figures that we produce. So the top half is the main paper, bottom half is the supplement. So this is something that I'm not the first person to observe this, but scientists, it's really important to have figures in your paper. Like a lot of the storytelling happens through the figures. And the benefit for me as a philosopher of doing this, so I produce almost all of the data that's in here. There's a little bit that Aaron produced, and I drafted all of the paper and spent a lot of time trying to figure out how to present it. And the great thing about this was that I had this then like personal archive of like all of the stuff, right? Like it was very tiring about what happened. It's like, okay, when was this experiment done? What figure did it go to? How many figures did it contribute data to? How many versions did it go through? What went into the supplement? What didn't? So I did a lot of things with this, playing around, visualizing it in different ways. I think the most interesting thing that I ended up with was this graph. Okay, I think you can see almost everything. This says order of experimentation here. Okay. So this is now just focusing on the main paper, sort of looking at the distribution of data. It's about 120 experiments, 130 that I did over the course of several years. The numbers here, 1, 2, 3, 4, 5, 6, 7, 8. Those are figures in the paper. Figure 3 was just a diagram, so it didn't have any data in it. Okay, so we had 8 data figures. So we narrate the paper in the order of this. So yeah, like this little pink bar is showing you one experiment here. Each of these little like short black bars are what I'm just calling data packets, for want of a better word, just some collection of measurements that went from that experiment into a particular figure. So here in this like first pink line, you can see there's like 4 data packets that this experiment contributed to the figures, and they're all different. They're all different bits of data, but they were all measured during the experiment, and they went sort of 4 different places in the paper. Okay, so that's that vertical direction. The horizontal direction, right, is each of these lines is one figure, like I said. This is the order of experimentation. So you can see, like, there's not a linear relationship here between like when we did the experiments and when they showed up in the paper. So an easy way to see this is with figure 8 here. This is like a little collection of experiments that I did when I first joined the lab, where we were looking at doing this procedure over multiple generations. Like, basically do the babies of worms who've been stressed produce babies that are better able to survive stress when they grow up. And in fact, they do. But the experiments were really, really hard to do. And so we didn't do that many of them. But they were kind of like a nice capstone to the whole narrative. So we stuck them at the end of the paper. Any questions about how this is representing? Tracking? Okay. So nonlinear, I think, oh yeah.
So when one experiment contributes to different figures, what
Yeah.
are the different aspects of another experiment that go into the different figures?
Yeah, so just like in this case, in this kind of situation where I don't have the right slide, sorry, I won't go all the way back, but like when we take them out, we would count their eggs, like how many eggs did they lay during stress, and then we'd count the live offspring. Those would be an example of like two different aspects of the experiment. So the data about like how many eggs were laid might go in like one figure or one panel, and the data about how many live offspring produced would go into another figure.
And each of those is a packet.
Yeah, so each of those would be a packet, right? Like, okay, in experiment 1, like 30 out of 50 worms like recovered live offspring or something. That would be like a packet of data. Right. So I mentioned the nonlinearity. The other thing I just want to mention from here is that we were pretty efficient in using data. Like basically, if there was any measurement from an experiment that we could use as part of the story, we did. And when we started this, we had some sense of what was going to be relevant, but I think there was also a lot of like, all right, like what do we possibly have that we could use to amplify the narrative in one way or another? And I think this kind of supports Hacking's point. Like experiments are hard, phenomena are hard, right? And you want to use them as best you can. So I suspect, that these features of sort of nonlinearity and efficient data use, when possible, are present to varying degrees in other studies besides. All right, so that's the end of that particular narrative. I wouldn't say I quite have a conclusion yet, but I thought a denouement, which are a few things that I have observed that I think might be interesting and generalizable, and I would like to know what you think. Okay, so possible generalizations here from this story that I have. First of all, this act of narrative construction, which I want to call creative, shapes science at multiple times, right? So I think we're sort of constantly trying to understand how our results hang together in stories that will make sense to other people and will illuminate the interests that we have about the things that we are studying, and that this happens at multiple times. And I don't think, as I sort of gestured at earlier, I don't think the creativity is totally inscrutable, but it involves a lot of getting your hands dirty and getting. The next point that I suspect generalizes is this iterative relationship between research questions, methods, and focal phenomena. I think you kind of start out with a question, but there's a lot of different ways you might pursue that question. Once you pursue it with a particular method, there's actually like a lot of different claims you might be able to make from the data that you get from that method. These are all so there's a lot of decision points, and then how the different decisions are made affect how other decisions are made, and there's this interplay that happens. I mentioned this point earlier, but I think data journeys take place on small scales too, and that's something that it might be interesting to think more about. Like, how are these small scale data journeys similar to or different from the ones that involve much larger crossing of time and space? Then the last one kind of generalizes, particularly for my case study, these phenomena may often be multiply specifiable. So not gesturing at the language of multiple realizability, but it's something different here, right? Like you have a phenomena like, male signaling or male priming of the reproductive system or reproductive stress response. You can make that specific in a number of different ways. And sometimes that decision about how to make it specific remains open for surprisingly long in the course of the research study. So those are the things I came up with. I'm curious to know if others see things that I missed. But for now, I will wrap it up. Thank lots of people who helped me, especially my coworkers in the lab who were really patient with the philosopher and all of the questions. And then thanks to all of you for listening.
Right, we take a 2-minute break and we have plenty of time for questions.